Guys, welcome to another A-level biology video. Mr Phillips. Today we're going to be looking at the control of blood glucose concentration, which is obviously mainly controlled in the pancreas. And it's the pancreas that detects the change in blood glucose concentration. It's nothing to do with the brain. So let's have a look then. So first of all, you need to understand, well, where do we get glucose from? So obviously glucose is absorbed from the gut, following the small, from the ileum in the small intestine. And then obviously you can break down glycogen in the liver or muscle cells, which is a hydrolysis reaction. And you can also do glucogenesis, which is when you turn other molecules like amino acids and lipids into glucose, which we'll look at over this lesson. But the supply and demand vary greatly, so we have to have control mechanisms in order to make sure our glucose levels are correct. The pancreas, you should know, is this small organ, sort of here. Well, not so a small organ, but this organ here that sort of fits just underneath your stomach and liver, above your small intestines. Your pancreas has two functions. Okay, so it's got two functions. One is an endocrine function. Okay, and that is endocrine glands secrete hormones, as you should know, and that's what we'll be looking at in this lesson. But we've also done about digestion in the past. And digestion is what we call an exocrine function. So when it's releasing enzymes, it always releases enzymes into a duct, and that is into the small intestines, and that's an exocrine function. When it's an endocrine function, it is releasing hormones directly into the bloodstream, and the two hormones that the pancreas releases are insulin and glucagon, as we'll be talking about today. What about the pancreas? Well, the pancreas is made up of tissues called the islets of Langerhans, okay, and they're made up of two cell types. So if you're asked to name the tissues that make up the pancreas, it's the islets of Langerhans. If you're asked to name the cell types, it's alpha and beta cells. And you need to know the difference between these two cells. So first of all, we've got the tissue called the islets of Langerhans. Let's have a look a bit closer. The two cells, we've got alpha cells, okay, and the alpha cells are the larger cells, hence they're called the alpha cells. And they contain the hormone glucagon, and they secrete glucagon. The beta cells, which stay in this sort of dark red color, are much smaller, but they secrete insulin. And insulin is the thing that makes them go dark, because when it gets stained, it goes dark. Okay, so you might get a photo like this and be asked to identify them. So the darker red stain ones are the beta cells, or you might get something where you can see the different size of the cells, and the smaller cells are the beta cells. The beta cells release insulin, and the alpha cells, the larger alpha cells, release glucagon. And here you can see just a different version of them. Again, just showing you that they make up a tissue together. And the beta cells here are the smaller cells identified in yellow, but the alpha cells are the blue ones on the outside. Just again, definitely defining how they would differ in their staining. So how do each cell work? Well, alpha cells release glucagon, and you should know, hopefully for GCSE, that glucagon is when the glucose has gone. So therefore it raises the blood sugar level, okay? It's sensitive to low levels of blood in the low levels of glucose in the blood. So it's your alpha cells that respond to that. Your beta cells respond to when there is too much glucose in the blood, and it releases insulin to lower that glucose level. And that helps maintain a range between the two. So you should have about 90 milligrams of glucose in every 100 centimeters cubed of blood, and that's the level that our body sort of aims for. Okay. Now, obviously, if it goes wrong, it can cause significant health issues. So if you've got too little glucose in the blood, you obviously might run out of glucose, which is a respiratory substrate, and therefore you can't carry out normal activities. And particularly important in the brain cells, which only respire with glucose. So glucose is known as the chief respiratory pigment, but some molecules can only use glucose as a result. And if your glucose gets too low, it can also affect the osmotic potential of the blood, and that might result in low blood pressure. Now, if obviously then the opposite happens and your blood level gets too high, it could cause damage to the cell. And the main reason it causes damage to the cell is because it will lower the water potential, and that could cause water to rush in via osmosis. And obviously in animal cells, that's particularly dangerous because that could cause the cell to burst as a result. Okay, so this graph sort of shows you a summary of how blood glucose levels are controlled. So if you follow my sort of red line on here, once you've eaten, your blood sugar levels will increase, and that will be detected by the receptors, i.e. the alpha and beta cell receptors in the pancreas, and that then causes the release of insulin. So the alpha cells will stop releasing glucagon and the beta cells will start releasing insulin. They will then go to the liver, and the liver will respond by using by doing the things we're going to talk about. So the liver will do stuff like more respiration. It will convert glucose into glycogen. We'll talk about it in a few moments. Then your blood sugar levels drop down, and then there's a reverse because again we have to stop it getting too low. So then alpha cells detect that, start releasing glucagon. Beta cells stop releasing insulin, and that goes to the liver, and the receptors in the liver detect that, and they reverse what they're doing. So they'll slow down respiration, for example. They'll turn glycogen into glucagon. So that is an example of negative feedback, because what is happening is there's a change in the body, and then your release of insulin or glucagon is reversing the change in the body, taking it back to that normal level. But let's have a look at that in a bit more detail. So if we want to get rid of blood sugar, okay, so if we've got too much glucose in the blood, we will release insulin from the beta cells, okay, and we'll stop releasing glucagon from the alpha cells. That insulin will then attach to the receptors on the muscle cell or the liver cell membranes, okay, and therefore, so it's on the plasma membranes of the liver cell and muscle cell, and their receptors, which will be glycoproteins, glycolipids, and that will cause a response, okay. Now, there's basically 4 things that will happen as a result of that. So the first thing is there'll be an increased rate of facilitated diffusion into the muscle and fat cells, okay, increasing the number of channel proteins. We'll talk about that in a bit more detail in a minute. So that will help the glucose leave the blood and go into the liver or the muscle cells. We then also activate enzymes which do something called glycogenesis. And glycogenesis is where we turn soluble, which is why it's a problem, glucose, into glycogen, which is insoluble. Now that's really important. The fact that it's insoluble means it will not have an effect on osmosis, and therefore it won't cause damage of your cells that have lots of glycogen in it. That's how all our muscle cells and liver cells store glucose away. We then increase the rate of respiration, okay, and that obviously is going to use glucose because it's the chief respiratory substrate, and it's going to make sure that glucose is being used up, so that will help. And then we also increase the fat cell uptake by increasing lipid synthesis. What we're saying here is we can convert that glucose into fats, and then the fat cells can uptake them as a result, and that will lower the amount in the bloodstream. So a couple of extra bits here. Well, how do they do it? How do we actually lower the blood sugar level by increasing this uptake from the blood? Well, as you can see here, we've got a couple of stages in the diagram. Basically, the insulin molecule binds to the receptor on the plasma membrane or the cell surface membrane of the liver cell or the muscle cells, okay, and that then causes, and this is really muscle cells and fat cells, I should just say, not liver cells, that then causes a chemical signal to go into the cell, and in the cell there are vesicles that contain these special glucose carrier proteins, which is channel proteins effectively. And these then, when that chemical message comes in due to the attachment of insulin, these then fuse with the cell surface membrane and increase the number of channels in the membrane. Those channels are called GLUT4 channels. So that increases then your facilitated diffusion of glucose, which is obviously high naturally in the bloodstream at this point, into the muscle cell or the fat cell. So adipose is just another word for fat cells. Okay, so the next thing then is glycogenesis happens, which means glyco is a glycogen, okay, or glycogen is actually the whole word in there, but genesis means making, okay. So what happens then effectively is, as we just said, the glucose now has been able to diffuse in, because we've got these GLUT4 vesicles formed with the membrane, which is allowing the molecules in. And now we need to get enzymes, then get activated, which turn the glucose into glycose. So the insulin activates an enzyme called glucokinase, and that converts glucose into glucose phosphate, okay, and then that then allows it to convert into glycogen, but you don't need to know the stages of that, okay. And then the last bit, fat cells, enzymes will also then convert glucose into fatty acids and glycerol, which is how they do that. So what you need to just know is the insulin by attaching to the cell plasma receptors will cause the enzymes to be activated that do that conversion of glucose into glycogen, i.e. glycogenesis, and also glucose into stuff like fats and amino acids. So then the other side of that is what after a meal. So a healthy person after a meal, if they're going to eat for a long time, the blood sugar levels might drop, and you need to increase the blood sugar levels in your body. So how do you do that? Well, you do that by the alpha cells then need to secrete insulin, which obviously binds to receptors on the liver cell surface membrane. The beta cells will stop producing insulin as a result. And basically, one of three things happen. You've got glycogenolysis, okay, so it's glycogen, if you think of it like that, and lysis, so splitting of the glycogen molecule, which is just a long chain of beta glucose molecules. So you have a hydrolysis reaction happening, and that turns glycogen back into glucose, so it can be released into the blood. Glucogenesis, okay, is where basically enzymes are activated to convert amino acids and fats back into glucose, and also the rate of respiration will now fall as a result of stop using up that glucose. So all of this is a negative feedback situation because it's a mechanism that is used to restore the constant level that we should have, so it takes it back to a normal level. Now, your adrenal glands are involved in this as well. So your adrenal glands sit above the kidneys, and they obviously release the adrenaline hormone, which is involved in your flight and fight response, and it's involved in like your heart rate and things like that. But it's also, because you need glucose for the flight and fight response, it also controls blood sugar levels as well. So what happens? Well, what happens is when the adrenaline is secreted, it will be secreted when, in several situations, so stress and exercise, obviously, so we've said that before, that's when your adrenaline is released primarily. But we're not talking about that today. What we're talking about is when your blood glucose levels are low, that is when adrenaline is released, and it binds to the liver receptors. Okay, so its target cell is the liver, so it goes through the blood from the adrenal glands to the liver. This then activates glycogenolysis, so it activates the breakdown of glycogen back into glucose to increase blood sugar levels, and it inhibits glycogenesis, which means that there's no glucose being turned into glycogen. And it also activates glucagon release to insulate, and it inhibits, insulin release. So it will activate that glucagon release from the alpha cells and inhibit the insulin release from the beta cells. And this really is happening in muscle cells. It does happen in the liver as well. So that's important to know. So what is actually happening here? Well, you can see this is what sort of happens, and this links back to the hormone lesson on first and secondary messengers. The adrenal gland comes in, it binds to the receptor inside the liver cell surface membrane. That then activates something called a G protein, which you don't need to particularly know the name G protein. Okay, the G protein then activates an enzyme which converts ATP into cyclic AMP, and that converts glycogen into glucose, and that's where your glucose comes in. So in this case, linking back to the last lesson, the first messenger is your adrenaline, because that is on the one side of the membrane. That then triggers the reaction across the cell surface membrane, and your secondary messenger is cyclic AMP, which is triggering the change or the response as a result. Okay, so I hope that's been useful for you guys, and we'll continue to look at the control of blood sugar levels and what goes wrong with diabetics next time.